then talk about the applications. And
thanks, Joanna, and thanks, Holden. So I'm an instructor in oncology, so being a cancer doctor, I'm going to tell you some of how I've used this specifically to analyze specimens from my cancer patients and give you an overview, and in the process answer some of the questions that have come up. So as an oncologist, there are periods of opportunity that we can make assessments to actually make a decision for a patient. At some point, something happens and a tumor is initiated. It grows until there's some symptom that leads to diagnosis and staging. And this is the point when the doctors get involved. For lymphoma, because I'm a lymphoma doctor, the standard evaluation is you do a surgical biopsy, you get that lymph node out, and you look at it under the microscope with histology, immunohistochemistry, flow cytometry is very standard in lymphoma. And you get an overall assessment of the tumor burden in the body. How many places is this tumor involving? You do CT imaging, PET scanning. And then at the research level, we're always trying to find better ways to make the diagnosis and figure out how well this person's going to do based on characteristics of the tumor. We're really great at looking at DNA now with karyotype sequencing and deep sequencing. RNAs, microRNAs, again, very sensitive quantitative PCR, microarrays. But where it's been more difficult has been at the protein level because of, as Joanna said, the amount of tissue that you really need to make a lot of measurements. And especially when it comes to phosphorylation. So the Western blot takes up to 1 million cells. You know, for flow cytometry, you've got to start with some minimum number in your tube. And even mass spec requires at least 1,000, 10,000 cells. And don't forget, anything that you find either with RNA or DNA really needs to be validated at the protein and phosphoprotein level because that's really where everything happens. A lot of our treatments now are targeting protein kinases, and you're only going to see changes in phosphorylation. Looking for a change in RNA is not really a direct measure, and certainly the DNA is not going to change. And then we choose our best treatment based on our best tests, and then right now we wait up to 3 months of giving a treatment, and then we evaluate for clinical response. And how do we measure for this response? We simply reevaluate the tumor burden. Has it shrunk or not with a CT scan or a PET scan? Is it less hypermetabolic? That's it. We really don't get information about, did the drug get to the tumor? Did it actually hit the target we thought it was supposed to be hitting? If it did the cells actually apoptose? We're really great at this in mice, but in people, we really don't know. And that's what I call the period of biologic response. Because the day you start a drug, things start happening, right? It gets metabolized, it gets to the tumor. But we really don't know what's happening. All our measurements now are just getting a CT scan to see if the tumor has shrunk. So this is where NanoPro has come in really useful. I use it to profile changes in cellular pathways of tumor cells that occur shortly after administering a therapy. Instead of waiting 3 months, I'm talking like day 8, day 3. We measure a biologic response, which has something to do with the target and the microenvironment. And this has been difficult historically, like I said, because of the large amount of a specimen that would be required to do it by the gold standard, which is Western blot, and the processing requirements to do this. Lack of highly quantitative methods. Everybody's done a Western blot, and we know how variable that can be depending on how long you stained and even well-to-well variation. And it's been very hard to measure complex phosphorylations, which you'll see in the next slide. So this is where I've used NanoPro, which I abbreviate as NIA, nanoimmunoassay, for both charge-based separation, and there's a newer version where we can do size-based separation as well. And I've been using them to profile cancer signaling pathways in patient cells. So just as one example, in the preclinical, in the Felcher lab, we found that Lipitor, a cholesterol-lowering agent, can cause apoptosis in lymphoma by inactivating the RAS and MYC pathways. And that's a published paper. I'm not going to go through it. But basically, it's associated with decreased MAP kinase inhibition, decreased MAP kinase signaling, as well as decreased STAT3 phosphorylation. So I wrote a clinical trial to test this drug, Lipitor, a cholesterol-lowering agent, as a single agent to treat lymphoma. And what I built into the study is this thing that nobody else can do. You take a sample of the lymphoma by a fine needle aspirate pre-therapy, sample again after 8 days of treatment, and I'm looking for these molecular changes that we saw in the mice, decreases in STAT signaling, decreases in MAP kinase, and then we do our standard stuff at 3 months and see if it all correlates. So this is the first published example we have of this, where we took specimens from the patient prior to treatment, and you can see it's not necessarily a clean signal, and we validated which peaks, which I'll get into in a minute, are specific for phospho-STAT3 and STAT5. And day 8, even when you normalize to the amount of input with the loading control, we can see that phospho-STAT3 and STAT5 significantly decreased. And the error bars don't overlap. It's very specific. Now, in contrast to flow cytometry, I have the luxury in lymphoma. Basically, it's a single cell suspension when you do a fine needle aspirate. I took half of the specimen and did flow cytometry on it too. And again, you can see that day 1, if we normalize to day 1, day 8, there's a very, very subtle change in phosphostat-3 and STAT5 signaling using the exact same antibodies. But you can see I cranked up the scale from -1 to 1, so it's a really, really subtle change. And then day 43, when we sample again, the change is, it's kind of gotten used to it. But interestingly, flow, you can get single cell resolution, right? You can gate on the different populations. So if we gate on the tumor cells themselves, which are CD20 positive, you can see that there's a decrease in the signaling in those cells, but in the CD20 negative cells, there's an increase in signaling, which starts to hint that there's actually some interaction between the tumor cells and a differential effect on tumor versus non-tumor cells. So this is an example how you can use the data together. That nanoimmunoassay showed that there's definitely a decrease in phosphostat-3 and 5, but flow could give you a sense of what the different populations are. But what flow couldn't show us is quantifying the decreases in phospho-ERK. If we looked at ERK, which Joanna had showed you several examples of the traces, it's just not sensitive enough to see the changes. But because NanoPro is incredibly quantitative and sensitive over 6 logs, we could get very specific, significant changes in all the different isoforms. So maybe I'll take a second now and just answer how we know that it's monophospho-ERK1 and for ERK2. So part of assay development, and this is what Tram is working on, and I've spent years doing this, is one, determining which peaks are specific. So it's like a Western blot. You do block steps, you do wash steps between all the antibody washes. But with ERK, for example, we have the luxury of having lots of different antibodies. So if you run ERK1, you get a certain number of peaks. If you run ERK2, you get a certain number of peaks and see which ones overlap. You treat with phosphatase, the phosphopeaks go away. So you go through great lengths to validate every peak and see what's specific. The data that you get once you've done all that is, I think one thing that didn't really come through is it's so high resolution that even a monophosphorylation can be measured. So when I say p-ERK1, I literally mean monophosphorylated ERK1. You guys might know that there's 2 phosphosites on ERK1 and also 2 phosphosites on ERK2. And there's no antibody that's specific to the monophosphoform. And I've called cell signaling and begged them to do it, but they only have one that recognize both phosphosites. So you can't see that data with any other technology except if you ran a mass spec, perhaps. Okay, and so on that point of monophosphorylated isoforms that hadn't been measured before, we also found something really interesting in patients with CML, chronic myelogenous leukemia, treated with imatinib, which is one of the first known targeted therapeutics in cancer. It's a drug that inhibits the kinase, tyrosine kinase BCR-ABL, which is activated in these patients with leukemia. And it's been published by many people before that you turn off the dual ERK2 phosphorylation when you treat these patients. But what we found by NanoPro is that there's that one peak that changes in cells from patients, drawn directly from patients that have been treated. And much to our surprise, it was a monophospho-ERK2 isoform. There's very little phosphorylation of other forms. And when you use the traditional phosphoantibodies in Western, it looks like this, but you didn't know it was monophosphorylated. The other point is that you tend to think there's a lot of phosphorylation basally, and then when you're inhibiting tyrosine kinase pathways, you're turning them off a lot. Well, here, it was really surprising to find that pre-treatment, it was only 18.5% phosphorylated. Getting the relative phosphorylation, how much is unphosphorylated and phosphorylated, is also something that's really easy with this technology and almost impossible with others, even with phospho-specific antibodies. So when we look across a panel of patients with CML, if you look at blue, these are the untreated time points, and then we took a serial sample from the same patient once they were treated. This is not taking cells out and treating them in a dish. This is taking the cells out of the patient at different time points and running them immediately. And what you can see is there's a large range of basal expression. The blue looks like it ranges anywhere from 0 to 21% phosphorylation. But what all the patients had in common that responded to the treatment is they were able to inactivate the pathway. You can see a significant difference between the blue and green for each patient. The patients that were relapsed or refractory had lower levels to begin with, but they weren't able to inactivate the pathway any further. And so I think this is going to be a much more powerful way of predicting who's really responding, is if you can get a sample early during treatment and see if you're actually turning off the pathway and to what extent. Because a lot of people are just looking at the basal time point, the blue time points, and trying to set some threshold and say, oh, everybody who's got a high level is going to respond and people with low levels won't. But that's really not good enough because there's very different basal signaling, and it's really about the ability to turn down the signaling pathway. So that was a major learning point for me as an oncologist. So I've shown one example where there's a specific monophosphoisoform that can predict outcome to treatment. Is this just a one-off in CML and imatinib, or is it possible in other therapeutics? So we've started to look at potential biomarkers in other preclinical and early clinical trials. And I'll just give you two more examples. One, this is a brand new drug. Bryostatin is a natural product that comes from the sea sponge, but it takes 2 tons of sea sponge to get 1 gram of clinical material. Had a lot of really exciting activity in lymphomas, but we ran out of drug and stopped the clinical trials because we couldn't keep going. So Paul Wender's laboratory here at Stanford has spent decades synthesizing analogs to this, and their lead compound we put first in mouse, and they call it Picolog. And this acts by activating the protein kinase C pathway, so you'd expect MEK phosphorylation to go up. So when we treated mice with these, you can see that prior to treatment, there's a certain level of phosphorylation. But once you treat with Picolog, there's a preferential increase in phosphorylation of one of the isoforms. So again, another hint that we're actually activating drugs, or drugs are inhibiting pathways in a very specific manner that we just couldn't see before. And I'll give one last example. This is an example of Regersitib, a novel multi-kinase inhibitor currently in phase 2 and phase 3 trials. We did this in patients with myelodysplastic syndrome, enrolled 13 patients, and we got serial bone marrow samples from them. We were looking at this rare CD34 stem cell population to see what happened when we treated these abnormal stem cells in the patients with this drug. And this is an example of some of the data that we got from CD34 bone marrow samples prior to and post cycle 1 of treatment with the drug on a clinical trial. And you can see that the major change is in these 1, 2, and 3, and 4 peaks, which are all phosphorylated isoforms of AKT2, which is downstream of PI3 kinase. And what we found is that, again, the phosphorylation doesn't completely go away. There's, at maximum, we saw a 30% decrease, and we could specifically see which of the isoforms were most predictive, and it seems to be isoform 3 has the most. So when we looked at isoform 3 and phospho-AKT2 for that matter across the panel of patients that we could get serial samples, all of those that had a good clinical response, either the bone marrow cleared of blasts or it didn't increase in blasts, we saw that the isoform 3 was able to be inhibited. Those patients that had progressive disease, that particular isoform and AKT2 phosphorylation stayed the same or increased. So again, another hint that if we're able to look early on during treatment, we can start figuring out which isoforms might be part of the mechanism of activity. And so we're now extending these studies, that was in a phase 2, now to a phase 3, including a Stanford site. So I know it was fast, but I'm happy to answer questions. Basically what I've said is that immunodetection of proteins, both, I didn't show you size-based, but we can do it with size-based and charge-based NIA, are enabling highly quantitative profiling of patient tumor cells before and during treatment. And I've shown you examples in cancer, but you can easily imagine how this could be done with different immune cells that you've sorted and things like that, or stem cells, or lots of different applications. Profiles of individual protein isoforms and their relative abundance can be used to interrogate the degree of biologic response. I'll give you an example of atorvastatin in lymphoma, imatinib in CML, regersitib in MDS. And in preclinical work in mice, these are easy. I mean, I never do Western blots anymore. I just load these plates, and you come back the next morning, you've got your answer. But in the clinical translational work, where I'm working with a really limited number of patient cells, this is making it possible to do these studies.